Really? Okay. Well, afternoon, everybody. It's now noon, and I'm Desmond Schatz, and it gives me really great pleasure to introduce a friend and a colleague and just a truly outstanding scientist. This is a real treat for me. It's a treat for us. And it's not only a treat because of his science and his professionalism, but as a human being and what he stands for in terms of understanding the world and the search for truth. And that often goes beyond science. So I've gotten to know Yuval really over the past, I would say, 15 to 20 years as a scientist, as a friend. And he is a Ph.D. Who's really devoted his life to really understanding organ size, the cellular origins of tissues, tissue homeostasis, and the molecular underpinnings therein. We've had grants looking at the death of beta cells, as well now as trying to understand, why insulin is not only not working, why are beta cells dying, and how we can restore insulin production in patients with type 1 diabetes. He is heavily cited. He has multiple grants from multiple institutions and has, again, many publications. So really, it gives me great pleasure. I wish that he was staying here for longer. Some of the students had an opportunity to meet with him. He has promised that he will be coming back in a year. So next time it will be certainly for much longer. But again, people didn't come to hear me. They came to hear the speaker. And it's my pleasure to introduce Yuval Dor.
Thank you, Des. After all this introduction, as they say, I wonder what I have to say now. So thanks a lot. Very nice to be here for the first time. It'll be a pleasure. My understanding is that there are people at Zoom, so I should probably use, instead of a mouse, do you see when I? No. Well, so I'll just. So yeah, okay, I'll point to this and they'll understand. Anyway, so I'm really glad to be here, and I'm here to tell you a story about something that is still unpublished. We just put it in bioRxiv. Story about the stories RNA editing deficiency as a potential route to diabetes, maybe type 1 diabetes. And this is a collaboration between my lab, mostly led by a fellow called Agnieszka Chandler, and several MD PhD students, mostly, with a computational biologist at Bar-Ilan University, Erez Lebanon, and his student. They're really experts in RNA editing, which I'll tell you about. Collaboration with Al Powers and Chulna Dai, which many of you probably will know from Vanderbilt. Klaus Kessner from UPenn, and with my dear friends here at UF on the specific aspect that I'll be talking about as well. And always we're collaborating with Ben Glazer, the head of endocrinology at Hadassah, and these are funding agencies. So I'll start with this chart, which is probably familiar to most of you, and that's a redrawing of the classical chart of George Eisenbart, which describes how we see. To date, I think the pathogenesis of type 1 diabetes described as beta cell mass as a function of time. And the general concept is that you start with genetic predisposition, where at some point there's an environmental trigger, something happens that starts beta cell damage and an immune attack. We'll talk about that. And then starts a long period of so-called silent beta cell loss. We don't know that because we don't see that because there's functional redundancy. Until a few months or years later, there's clinical onset. And a major issue is, what is the nature of that trigger? So it's sometimes called an environmental trigger, sometimes called the precipitating event. There's this TEDdy consortium that is trying to figure out what this is. We don't know what it is, despite many suggestions. I think two hints that I want to start with are that there's clear evidence for an interferon response in early stages of type 1 diabetes, interferon, which is biologically meant to be an antiviral response mostly in islets as well as in blood. And another crucial hint is that GWAS studies have identified SNPs in a gene called EphH1 or MDA5, which is a double-stranded RNA sensor. Double-stranded RNA is a hallmark of the presence of viruses. So it turns out that mutations in that, or genetic variations in that sensor either protect or predispose to type 1 diabetes, further supporting, people used to say supporting viral etiology, but supporting the importance of double-stranded RNA. Yeah, I think this actually says the same thing, but with some key citations for the fact that T1D is preceded by this transcriptional signature, both in the periphery as well as in islets. And the second, which is really telling, is the classical science paper by John Todd, but then reproduced by many other people, the fact that the SNPs in this double-stranded RNA sensor are modifying the risk for insulin. So based on this and lots of other lines of evidence, I think a prevailing model for the pathogenesis of type 1 diabetes is that it all starts with a virus. Viruses produce invariably double-stranded RNA molecules. This is an evolutionary conserved dangerous signal telling the cell that there's a virus. This is sensed by this guy, EphH1 and MDA5, which activates a signal transduction pathway leading to transcription and secretion of type 1 interferon. This goes outside and either autocrinically or paraclinically would activate or lead to the transcription of interferon-stimulated genes. This will attract inflammatory cells, islet inflammation, and on the right genetic background, HLA mutations, etc., may progress to autoimmune type 1 diabetes. So this is the prevailing model. But there are problems with that. I think it's a controversial statement, but I think fair to say, particularly in this place, decades of search, we still have no definitive causal virus, right? We search and we're still searching, but no solid evidence that viral infection does lead to type 1 diabetes. The second kind of less known line of evidence that I think is really crucial is that when people deleted this double-stranded RNA sensor in NOD mice, right, the classical animal model of autoimmune diabetes that develops autoimmunity in SPF conditions, super clean conditions, no viruses there, right? It also reduces diabetes in that model. So this suggests that the requirement for this double-stranded RNA sensor in this super clean context of T1D suggests that the double-stranded RNA sensing is involved in autoimmune diabetes, even when there's no virus. This was the idea, and this is one of the studies that showed that. So we started by asking, can EphH1, which is needed, and the interferon response, which is present, can these guys be activated by endogenous rather than viral double-stranded RNAs? So it turns out there is a pathway that can generate these, and so here's a crash course on a key process in molecular biology, a key post-transcriptional modification called RNA editing. RNA editing is a process whereby adenosines on RNA undergo deamination. The amine group is removed by an enzyme called ADAR, or adenosine deamination acting on RNA, right? And when you deaminate adenosine, you get an unusual base called inosine on the RNA. The thing is that inosine is sterically very similar to guanosine. So from the perspective of everybody who's looking at the RNA, what you did is that you changed adenosine to guanosine. Now, this has two potential consequences. One is that if this happened inside a coding region, right? So you get essentially an A to G mutation, and this can change coding. And there are actually cases where you get changing in the product, the protein product, because of that RNA editing. It's an important process, mostly in brain function. So that's one potential outcome of the presence of editing. And if there's no editing, then you will get different amino acid composition. But the other impact or consequence of RNA editing is that in cases when there's double-stranded RNA And in double-strand RNA, one of the base pairs would be A with U, right? Adenosine with uracil. If that A is deaminated to give you an I, which is read like a G, so this does not pair anymore, right? So you disrupt double-strand RNA structure. And it turns out that the second is the most important, prevalent, and important function of RNA editing. So first of all, what is the main substrate of ADAR? Turns out that the main substrate is endogenous double-strand RNA that is generated by retroelements. So there is this, 10% of our genome is those retroelements, those most frequent ALU elements, like defective retroviruses that were inserted millions of years ago. This is 10% of our genome, about 1 million such fragments. And in thousands of cases, inside the 3' UTR of expressed genes, there's an ALU element. And in thousands of cases, accidentally, there's another ALU element that was inserted into the same 3' UTR in an inverse orientation. So many such fragments that this happens in many cases. So when you have a gene that is expressed and there's an ALU, an inverted ALU, the consequence will be double-strand RNA. This is dangerous because this is the hallmark of viral infection. So each time such a gene is expressed, what you are going to get is massive interferon response, cell death, and really havoc, right? So it's crucial for a cell to maintain its ability to detect real viruses by a real viral double-strand RNA, but to prevent this. And it turns out that the key role of RNA editing is to dismantle such endogenous double-strand RNAs to prevent the activation of this dangerous immune response. And the logic is that when you are infected with a real virus, then in the cytoplasm, there will be viral double-strand RNA. This will trigger interferon response, right? With COVID and with influenza and with almost all viruses, this is what happens. But when endogenous genes, while still in the nucleus, are producing these dangerous endogenous double-strand RNA, this is where ADAR comes to play, deaminates those A's that stand versus the U's, and you get this destabilized RNA structure that does not activate interferon response. And this is now the consensus as of the last 5 years or so, that the central role of RNA editing is to prevent an antiviral response. So here is the hypothesis, right? The idea that maybe instead of that pathway for viral infection, double-strand RNA signaling all the way to interferon, inflammation, and T1D, the idea is that lower levels of RNA editing, presumably in beta cells, may trigger the same pathway, right? Interferon, inflammation, T1D. The nice, a priori thing about that model is that it's a model that can accommodate the whole set of antiviral responses known to happen in T1D, but without a virus, right? And the experiment is pretty straightforward. Went to mice, and the experiment was to really delete the ADAR genes in beta cells and to correct in mice, in vivo adult mice, and to characterize the phenotype with emphasis on both the inflammatory immune response as well as the beta cell phenotype. This is the genotype, right? So floxed ADAR with a lineage tracer. Every mutant cell that you'll see in a minute will be GFP positive permanently. And this is driven by an insulin promoter driving CRE-R. So it's a tamoxifen-dependent CRE recombinase. We inject tamoxifen, ADAR is deleted. So I'm going to describe now the phenotype. But before that, many of you would ask me at the end, are these results relevant at all to human type 1 diabetes? Which is a very legitimate concern. I wasn't so concerned for several years because we said, well, let's just play with these mice and with the hypothesis, and we generate a model that can be tested later. Let's generate a shelf product of how diabetes could develop. But turns out that down the road, about 4 years after we were starting that, this is the question, and turns out that really only about 6 months ago came out this amazing Nature paper from the group of Jean Billy Lee at Stanford. These are human geneticists. And briefly, what they found is that they identified slips in the human genome that are impairing RNA editing in nearby loci, in cis elements. So I'm not getting into the mechanism, but there are places in the genome where genetic variations will impair RNA editing and encourage the formation of, since the disrupted RNA editing, so there will be more double-strand RNA. And the big surprise was that when they asked, well, what are these SNPs that affect RNA editing, turns out that they overlap with SNPs that are known from GWAS studies to predispose to autoinflammatory diseases, including type 1 diabetes, right? So genetic variations that cause defects in RNA editing also predispose to T1D, which is, you know, in support of our
yeah. Some viewers cannot see the entire presentation and we are trying to load out.
Yeah, so, yeah. Let's see.
Can they hear me?
Yes.
So, because I don't need a slide now, just to say that, so that's a kind of support, or I wouldn't say confirmation, but support to the overall model from human genetics, but it's human genetics. So as they say, you know it's true, but you have no clue about the mechanism. So they don't know which, where did they say that? I don't know, you took my slide off, but they don't know what are the cell types that matter, and they know nothing about the mechanism. But there's now genetic evidence from humans that this pathway that I'm proposing might play a role in type 1 diabetes. Okay, shall I continue? Okay. So that's, you know, the small deviation, there is human relevance from genetics, not environment. I'll come back to environment in a minute. Now this please. Oh, literally dead. Where's the guy? It's frozen. I can't even.
Go ahead. Get this notification. And if we just click on this, I'll try to advance it.
Thank you. Sorry. So to summarize, here's the working hypothesis, right? The idea is that impaired RNA editing in beta cells, which we will generate now by genetic manipulation, but may be occurring normally in humans, causes the accumulation of endogenous, not viral double-strand RNAs, activating the sensor, signaling pathway, interferon stimulated genes, inflammation, T1, right? Phenotype of those mice, they quickly develop diabetes, hyperglycemia within about 2 weeks. Heterozygous do not. Why are they hyperglycemic? Because they lose their beta cells. You see here the staining for insulin, right? The islets, and then it's almost completely gone when we measure beta cell mass, which you see here, the percentage of insulin positive cells is dramatically going down, which explains why they're hyperglycemic. And that's, you know, for the professionals here, this is also quantification of GFP. So GFP labels the lineage traced beta cells, right? And you can see that GFP is gone as well, right? Suggesting that beta cells just die off. But note that the extent of decrease of insulin staining is more than the extent of decrease of GFP. What does that mean? This means that there are GFP positive cells, that is beta cells, genetically labeled beta cells that are insulin negative, right? And we'll come back to that in a second. So many beta cells really die off, but some of them are still there, just insulin negative, and we'll get back to that in a second. So that's the key phenotype. Why does that happen? This is accompanied by massive islet inflammation, so-called insulitis. We see in multiple degrees of severity. These are CD45 cells, and most after 12 days, most of the islets are severely inflamed. This correlates very well with the degree of diabetes. So every dot here is a mouse where we score the percentage of islets that are severely infiltrated. And this is a calculation of the degree of dysglycemia. And there's a straight line suggesting that it's the islet inflammation that is causing beta cell dysfunction. So let's summarize, as geneticists, what we can say with confidence, that ADAR normally prevents islet autoinflammation, and most likely inflammation that causes diabetes. Who are these cells that infiltrate those leukocytes? Mostly macrophages, right? But surprisingly for me, as a very humble non-immunologist, including lymphocytes, right? cells of the adaptive immunity, what are they doing here, right? This is not an autoimmune model. Shouldn't be any epitopes of something. Why would cytotoxic T cells and T cells and B cells enter those islets? Still an open question. We don't know, but it's interesting and rhymes with type 1 diabetes. So inflammation, heterogeneous inflammation. Some islets are infiltrated, some are not, and this is dynamic. I'll say something about that in the end. Why does this happen? We're pretty sure that this happens because the mutant beta cells trigger a massive interferon response. All right, so we sort GFP positive mutant beta cells, look at their transcriptome, and there's massive elevation of interferon stimulated genes, and the gene sets are interferon gamma, interferon alpha, etc. So this is probably the double. So ADAR deficiency, double-stranded RNA, type 1 interferon stimulated genes, islet inflammation, diabetes. We see the same in human islets. So our collaborators, Al Powers and Chunra, knocked down ADAR in human islets and also see a massive interferon response. So the same thing happens in islets, in human islets. So, okay, so I just said that, but this is right interim summary. Deletion of ADAR causes reduced RNA editing, accumulation of the endogenous double-stranded RNA. I haven't shown you the data for that, but we see that clearly. This leads to an interferon response, recruitment of innate and adaptive, surprisingly for me, adaptive immune cells to islets, beta cell death, diabetes. I want to share with you now a few more kind of data questions.
Yeah, if
Please
you
go
don't
ahead.
mind.
Yeah, of course.
Before you move on. I'm curious to know if you've done the same ADAR deficiency in immunodeficient animals to tease apart if the beta cells are dying from the acute
Excellent.
interferon response or
Excellent
if they're actually dying.
Excellent question. Processes are right now underway to RAG minus. Exactly. We eliminate T and B cells, and I think it would be big news. Rarely for an experiment in biology, either result would be wonderful, right? Either we generated a system that involves really autoimmunity driven by lymphocytes without any predisposition. These are ICR mice, or that we recapitulate, as you'll see, many aspects of type 1 diabetes without T and B cells, without any specific antigen specificity. So we'll know in 6 months. But these are complicated courses.
Yuval,
Yeah.
along those lines, do all the insulin-containing cells express YFP, or can you segregate, right? Along those lines of thinking, dedifferentiation, why are they not affected by an autoimmune or immune attack?
First of all, as with all pre systems, I mean, knockout is not complete. The majority of beta cells lose ADAR, some remain wild type, and I'll show data on that in a minute. What seems to be the key determinant is whether the islet is infiltrated or not. Like in type 1 diabetes, if you're infiltrated, you're done. If you remain clean of immune cells, despite the knockout, you continue to function. But your day will come because within 2 weeks, I mean, everything is destroyed, right? So there's something really interesting about the dynamics. All right, so observation number 1, and you'll see, I mean, the reason I chose those observations that there are always things that remind me of type 1 diabetes. So that was, a chance observation when this is what I showed you before. When you take a 4-week-old animal, mutant, you inject tamoxifen, within 2 weeks, you see massive, you see diabetes, right? You see the same exercise in 6-month-old animals. You delete with the same efficiency, ADAR, nothing happens, right? You open up these mice, in those young animals, you see massive insulitis. In the old guys that did not become diabetic, they also have no infiltration. So young mice are much more responsive, paralleling the severity of type 1 diabetes that is more characteristic young children and adults. We don't know the reason for that, but I love the parallel. Point number 2, what does it do to beta cell identity? So within the infiltrated islets, in the islets that are not yet infiltrated by immune cells, nothing happens. But in the islets that are surrounded by leukocytes, we see the following. So look at this green cell. That's by definition a beta cell, right? Was recombined, right? This cell has lost proinsulin as well as insulin. is alive, but has lost insulin completely. Nice. But the second one that I want to show is even more interesting. This red cell, right? That's a GFP negative cell. So it's a beta cell that was not touched by the other knockout. It's a wild type beta cell just sitting in this environment. And look at it. It's pro-insulin positive and completely insulin negative, right? We like that very much because these are cells that Clive Wasserfall and Mark and others have seen in human type 1 diabetes, the so-called empty or ghost beta cells that are pro-insulin positive but insulin negative. We don't understand that, but again, this is recapitulated here. The two hints that we have is that this is associated with loss of the prohormone convertase PC1-3. So it seems that the cytokines downregulate the prohormone convertase, and now you're stuck with pro-insulin that cannot be processed to functional insulin. And the second is we treat these with ruxolitinib, which is a JAK-STAT inhibitor, essentially preventing interferon signaling. This is reversed or prevented, suggesting that these are the interferon coming maybe from the infiltrating immune cells that is causing this, and maybe suggesting why this is happening in human T1D. Right, that was one. The other observation regarding beta cell biology is something that we completely don't understand. So I'm putting in here out for the first time ever for someone to help us out. This is a beautiful islet in a normal mouse. We know how to stain for insulin. It's clear and smooth and nice. Only in the islets, everything is good. In the mutants, in the infiltrated mutants, you very frequently see these white dots. This is white is the insulin. So this is only the insulin channel. You see, these are not within cells. These are extracellular aggregates of insulin. Many, many here. Another example from another mouse also. What are these? They are not inside cells. So I've never seen that before in any. Yes, Holger, you may know.
No, we also see this in stem cell derived cells in certain grapes.
Interesting. I don't know what it means, but it's, you know, standing out. I've seen dozens of mouse strains with diabetes. This I've never seen. There's something interesting, right? But we don't understand. And I think two more points, and then I'm mostly done with data. One is that, as I told you, most beta cells eventually die off. Look at the GFP here; it's almost gone. Here, it's starting to be gone. And in the context of massive infiltration, the red here, alpha cells and delta cells untouched. Nothing happens. Massive inflammation, infiltration, they're perfectly fine. So they don't care about that. So either there's something special about these cells, or you would say they're wild type, right? We deleted ADAR in only in beta cells. So we said, okay, let's knock out ADAR in alpha cells and see what happens. And we did that with the glucagon Cree-R animal that we got from Klaus Kastner. So now we knock out, it's a different mouse model now. We knock out ADAR specifically in alpha cells. Very efficient knockout. All alpha cells lose ADAR, become GFP positive. And when you look at their transcriptome, they completely lost RNA editing, and nothing happens, right? No phenotype at all. Alpha cells remain there, no inflammation, no metabolic phenotype as far as we can measure. Nothing happens. We don't understand that. Either, you know, we can hypothesize that either because they're so sparse, alpha cells, maybe you need a minimal number of, you know, adjacent knockout cells to talk with each other to trigger, you know, a positive feedback loop of interferon and inflammation. Another hypothesis that I actually prefer is that perhaps it so happened that the transcriptome of alpha cells does not normally produce those dangerous double-stranded RNA molecules, and then they don't care if there's no RNA editing. But then we did another experiment where we did a double knockout. So we knocked out ADAR in both alpha and beta cells. So now we have alpha and beta cells mutant for ADAR, and because of the beta cells, there's now massive infiltration. Beta cells are gone, alpha cells remain. They really don't care about inflammation, even if in the context of not having ADAR. There's something special about alpha cells, or maybe something special about beta cells, but there's a big difference. And again, this is a parallel to the selective elimination of beta cells that we see in T1D. Typically, we ascribe that to, you know, epitopes that are, you know, in beta cells and not on alpha cells, but here, no epitopes. So there's something more kind of inherent or innate. And my, I think, last point, few minutes now on maybe I think the most interesting or important observation is that we discovered that beta cell metabolism, specifically glycolysis, matters very much for the extent of the interferon response. So let's go over that experiment done in islets extracted from mice. So mice heterozygous for ADAR deletion have no interferon response. This is beta-2 microglobulin, but we see the same for all interferon stimulated genes. Nothing happens, just like a wild type, and they don't care which glucose levels you put them in. Knockout animals in low glucose produce no interferon response, but you put them in 11 millimolar of glucose, boom. So interferon response of those islets is massively affected by high glucose, or another way to put that, you need high glucose in order to produce that. It's actually, you don't need high glucose, you need glycolysis, because we can also treat with low glucose, we treat the islets with small molecule glucokinase activator, right? So in the insulin secretion pathway, if you boost glucokinase, then no matter what is the concentration of glucose, the beta cell sees, higher flux of glycolysis. So this is also boosting that. And turns out that the signaling pathway that moves from glucose or glycolysis to interferon inducible genes is going actually through the classical insulin secretion pathway, because if we treat on the background of 11 millimolar glucose, we treat with diazoxide, right? So this is a potassium channel opener that blocks that signaling. The signal is gone. If we give high glucose but block calcium channels with nifedipine, there's also no interferon signaling. So somehow glycolysis via calcium signaling is essential for interferon signaling in beta cells, which I think is crucial. I'll explain why in a minute. This is the most complicated experiment in the entire thing, and I'm almost done. We're trying to ask whether that effect of glucose matters in the originating cell, which sees double-stranded RNA and goes all the way down to interferon, or whether glucose matters in the cells that sense the secreted interferon and via this pathway induce those genes. Understand the two possibilities. So the way to distinguish between the role of glucose here or here was to take wild type islets, right? No double-stranded RNA, just wild type islets, put them with interferon with or without glucose, right? And when you do that experiment, so interferon in any case induces a burst of interferon stimulated genes. But if you do that in high glucose versus low glucose, you see always a twofold elevation. So the kind of slightly complicated conclusion from that is that glucose, to a large extent, pushes the interferon response of the original cell, the cell that has double-stranded RNA. We have a model why this happens, but to a lower extent also boosts the interferon response of a normal wild-type beta cell, which is what we have in our bodies. So this is really important. So, and why do I think this is really crucial? Because it suggests a potential vicious cycle that could drive diabetes. So, so this is how we see that. So let's imagine an injury to beta cells for some reason. I'm not even saying why, right? This will reduce insulin. This would reduce insulin secretion, right? Beta cells then have to work harder. So there's then higher workload in the beta cells. What will that do? According to what I just showed you, this will boost a pre-existing low but pre-existing interferon signaling, right? This will trigger interferon stimulated genes, which, as I showed you a few slides ago, then harm the beta cell by reducing insulin and etc. And this will further increase that. So that's a positive feedback loop that may take you from low-level injury interferon response to, really a fire. And further reasons why we're excited by that is that if probably all of you know of the honeymoon period, right? When after diagnosis you start treating with insulin, everything goes back to normal, right? There's recovery, beta cell function recovers. We typically think of the honeymoon period as a time where the metabolic load on a beta cell is reduced because you start treating with insulin. Maybe the honeymoon also has an immune component by reducing the burden on the beta cell, reducing glucose, maybe then also the interferon loop is reduced, which is interesting, and it's also connected to the classical notion of beta cell rest, which is known for decades that if, at some point, even in the early stage of type 1 diabetes, you force beta cell rest, you treat with the azoxide, for example, progression is slowed down. Typically interpreted as a lowered metabolic load or stress, maybe also lowered inflammatory milieu. And this, if true, this really immediately suggests approaches for prevention, for calming down the inflammatory environment. So we like that model very much. And the last data point, I think, is that inspired by that, Shani, the MD PhD student, went back to her miserable model of alpha cells where she knocks out ADAR and sees nothing. And she said, oh, maybe alpha cells also need a metabolic push. And when it turns out that when she treats islets with alpha cell knockout of ADAR with palmitate, then yes, she can extract an interferon response that is also inhibited by nifedipine. So, and this is really a partial story. I don't have anything to say beyond that. So in the case of beta cells, glucose is driving that. In vivo, this leads to islet inflammation. In the context of islet, if alpha cells deficient for ADAR, all I can say is that in vivo, nothing happens. And in vitro with palmitate, they are able to drive interferon signaling. How this can be translated in vivo, I still don't know the significance of that. But, so I think I'm almost done. So few kind of conclusion slides. Still okay, right? 2 more minutes. So in summary, what I showed you is that ADAR inactivation in beta cells is causing islet inflammation via the double-stranded RNA sensor EphH1. I haven't shown you the data, but when we, on top of the knockout of ADAR in beta cells, we also knock out EphH1, that sensor, then nothing happens. So all the phenotype that I showed you is strictly mediated to this double-stranded RNA sensor EphH1, which is important because ADA was reported to have also other functions. But in this case, everything goes through the EphH1. This leads to an interferon response, which drives islet inflammation, which drives beta cell specific dysfunction, followed by beta cell death, while alpha cells and delta cells are spared. This is all modulated by metabolism, glucose in the case of beta cells. And again, the fair, modest conclusion is that the key role of RNA editing in beta cells is to prevent islet inflammation. And 2 last slides that I want to collect all the interesting parallels that we noticed along the way between what we see here and human T1D. So in both human type 1 diabetes and this model, there is an interferon response that requires EphH1 that is followed by highly heterogeneous in temporally and spatially islet infiltration. Second, the phenotype is more severe in young age. We don't understand that in humans. We don't understand that in mice, but same or similar. In both situations, beta cell dysfunctions, such as reduced expression of insulin and other key beta cell genes, precedes beta cell death. This, surprising sparing of non-beta cells, and here it's in the context of something that should have been involving adaptive immune cells that are epitope targeting specific epitope. So as if the innate immune system is able to discriminate between alpha and beta cells. And the last thing is that the link between beta cell workload and accelerated progression. So to wrap it all up, the way we highly speculative model of T1D, going back to the model of pathogenesis, the way we speculate it now is that there's the genetic part of the story, which involves the predisposing HLA genes, which will affect your chance of getting autoimmunity, the predisposing EphH1 SNPs shown before to also affect probably the first stage of antiviral response, and now these new RNA editing SNPs that impact the accumulation of double-stranded RNA. And so that's the genetic part of things. And as for the environmental trigger, the speculative model is that there is this vicious cycle where I don't know where it starts, but at some point there is beta cell damage causing increased beta cell workload, which does two things. First, it will feed back to this or enhance interferon response, which will further harm the beta cell and will accelerate that loop. And the other thing is our leading model is that that beta cell dysfunction changes the transcriptional landscape of the beta cell. And when you change the way the RNA editing people think about that is that the transcriptional landscape of a cell will determine how many dangerous double-stranded RNA molecules are there. And so the thought is that the beta cell under glucose load would produce transcripts that produce double-stranded RNAs. And when RNA editing is defective, this will push that further. So two kind of mechanisms for a vicious cycle that make things worse. And that's it. I think happy to take questions. Thank you.
Thank you, Yuval. Let me just challenge you for a second. Is the interferon response, the chicken and the egg, dependent on glucose? Because in pre-diabetes, well before the appearance of antibodies, glucose levels are completely normal. Glucose levels are normal. And so we know in the honeymoon period, you even treat with insulin, or you treat it with insulin prior to diabetes, it does not prevent diabetes, which goes to an inherent defect within the beta cell. How are you going to show it? In other words, maybe it's not glucose, maybe it's lipids, maybe it's some other reactive oxygen species. Why are you so fixated, if you will, on glucose? We understand it's diabetes and glucose, but how are you going to show it?
So good question. And first of all, we tested glucose, but it is possible there are other metabolic injuries or stressors that will trigger that as well. But to your point about beta cell injury and the whole thing starting when glucose is normal, what you said is that glucose in blood is normal, but the beta cell workload, the glycolytic flux within the beta cell can be abnormal, right? For example, if you lose a third of your beta cells, glucose will be perfectly fine, but each beta cell will work 30% harder. So the flux, that won't be seen from the outside, but so an injury to the beta cell may trigger this abnormally high glucose metabolism without manifestation. Manifestation is when it's so bad that insulin produced is not able to compensate. But perfectly possible that other metabolic hits, maybe lipid metabolism defects. We actually haven't tested palmitate in beta cells, which might be actually a good idea. So all I'm saying is that the concept is that metabolic stress might cooperate with interferon signaling to start this vicious cycle. I'm absolutely convinced that there has to be a vicious cycle. You can't start with something and that's it. There has to be amplification. But
I have a question. Well, first of all, thank you very much for your presentation. It's very clear. And well, my question is similar to what Shaz, because we found that we could detect in daily results, we could detect not only the lipidic environment, but also the upregulation of reactive oxygen species proteins that are usually inside cells.
Yep.
And perhaps they are somehow involved in this situation in the islets. So perhaps there is an increase in ROS levels inside cells, and that could be also triggering the response that you are detecting. And oh yeah,
I fully accept. I'm on the same page in the sense that I also believe that the initiation of that thing of the whole process is beta cell injury. But there has to be an element that explains why the interferon response and the islet inflammation will be enhanced and enhanced and enhanced. And I think there must be, therefore, positive feedback loop where this beta cell injury calls upon the inflammation, which further accelerates that. And so that loop, and I think this is a putative mechanism for that loop where, because this was not mentioned before, that beta cell accelerated metabolic stress can further drive interferon and feedback. So that's the suggestion, which I think may apply even generally to the situation, even if RNA editing is not finally, turns out to be the key driver of type 1 diabetes. That metabolic driven loop, I think, is important by itself, because it immediately suggests also interventions, metformin and insulin, not insulin low level just to tolerate, but insulin that will reduce the burden on a beta cell. That was not tested. No, it doesn't like it.
Yeah, real quick, the other thing that we found that was very interesting is that we found that situation prior to 1 year prior to either immune, and this signaling of regulation was almost constantly during the whole period. So it's not. Well, we will think that it's not like a triggering, but it's like a constant
Pre-existing condition. I love that study. I think it's crucially important, and I'd love to be able to link it to what happens later on. But I think it's a very important observation that hints on what is really happening.
I'm not a fan of viruses, but this doesn't preclude the reason we haven't found a virus is because any virus could start this off.
That's true.
And so many different viruses. So trying to find an etiological virus will be impossible.
That's, we discussed that yesterday. It's a fundamental question in the field. Yes, there is this viral hypothesis. How conceptually will you convince yourself in the field that yes, T1D is caused by a virus, or no, T1D is not caused by a virus? There's not even an answer to this fundamental question. So what will it help? Suppose I find now another 100 cases with viruses. Does it prove anything? So we don't even have a way to distinguish, but I agree, almost any virus will have double-strand RNA, although viruses typically act for a short time.
But
So
it spins it up and then the feed forward.
could be, but defects in RNA editing are there, from the day you're generated. So that's maybe more attractive. But yes, I mean, too many people.
I do.
Yeah.
I do lots of calcium imaging, pancreas tissue slices.
Yeah.
Kind of see a lot the high glucose response. Like when people start to get multi-antibody positive, or people with type 1 diabetes.
Sure.
The other thing that you were talking about, sort of interferon response, kind of very closely tied to the calcium response associated with glucose processing in the beta cell.
Yep.
So I was wondering what you thought if, like, we kind of see a lot of that calcium response sometime prior to the onset, how that kind of fits, because there's also this loss of the extreme interferon response.
So what I showed mostly is that calcium influx is required for boosting interferon signaling. You're saying that, at early stages of T1D, calcium response is reduced, and the hint that we have from the transcriptome of those beta cells that are affected by the inflammatory milieu is that MAF-A is gone, PC1-3 is gone, so the beta cell, transcriptional functionality is gone, which then may explain why calcium channels eventually will be less represented. That would be my gut. Being there, I think.
Nice mouse work.
Thank you.
Quick question. Have you thinking about looking into like human samples and looking into ADA gene expression?
Yeah.
And that product? If you're interested, I know a place you can test.
All right. How much will it cost me? Yes, we've thought about that, and more than us, the reviewers of our grants and papers have thought about that. So at one level, as I mentioned, we collaborate with Al Powers, and what can be done with isolated human islets is perfectly recapitulating what I showed you here. You knock down ADAR, human beta cells, human islets respond just the same. How to go beyond that? ADAR itself is probably not, ADAR itself genetically is probably not playing a role. It's constitutively expressed. There are no variants of ADAR associated with T1D. There is a disease, a syndrome caused by ADAR deficiency called Aicardi-Goutier. Very severe disease. I mean, diabetes is there, but it's like very down the list of there are encephalopathies and wheelchair and cognitive. I mean, this is really devastating disease of massive autoinflammation. What this guy, Jim Billy Lee from Stanford, discovered that it's not ADAR that is defective, but it's actually the cis elements that prevent proper editing of sites that should be edited. How to test that on human materials such as NPOD, I really don't know. And
You don't need a knockout in human, so maybe you will see reduced activity or level in samples between control and type 1.
correct. So we invested efforts in trying to look for defective RNA editing in material from type 1 diabetes. Right. The problem is that up until very recently, the materials you could get from type 1 diabetes was like RNA sequencing data was messy. It was, composite of many cell types, and you can't figure out what's coming from the beta cell. As I think now with more precise, detailed single cell RNA sequencing data, we can go back and ask if there's evidence for that. But it's tough because the model, I mean, I'm complaining about the virus model, but it's also for this model. The model is very open-ended. I mean, the model may say that when you are born for 2 weeks, there's a defect in RNA editing, which then starts the fire, and it could be also transient. It's hard to test, but oh, nice. Thank you for joining. Oh no, that's I don't know who wrote that. If ADA1 is ADA1 the only known subtype of ADAR involved with regulating interferon response in beta cells? Is there an ADAR2, 3, etc. involved in the same process? I'm sorry. Yes, there are ADAR1, 2, 3. ADAR3 is a pseudogene. ADAR2 is mostly expressed in the brain and is responsible for coding and recoding. In beta cells, there's only ADAR1 that does this double-strand RNA disentanglement, and there's no compensation. There's a nice slide that I didn't show you. When you knock out ADAR, there's no compensation in either alpha or beta cells. So this is the one gene that matters for destabilizing double-strand RNAs and is the key player.
Great talk here.
Thank you.
In one of your conclusions, you mentioned metabolic activation of beta cells and alpha cells through glucose or palmitate.
Yep.
Amplifies interferon response. Can that be how long was your exposure to glucose or palmitate? Was it chronic? Because actually in real time, I see a decrease in or reduction in reactive oxygen species in real time upon exposure to glucose. So that was pretty interesting. You saw.
Interesting. These experiments were done with islets isolated from mutant or wild type islets, put in culture and exposed for 2 or 3 days. That's everything that I showed, and recently realized that there might be more hiding in the kinetics before. So what the students are now doing are taking time points of 2 hours, 10 hours, etc. So maybe there is interesting dynamics before so I suspect it's a kinetics of hours. It can't be less than that because what you test here is interferon stimulated genes. So you need type 1 interferon to be, activated, transcribed, translated, going out of the cell, inducing interferon stimulated genes elsewhere. So there has to be some lag, but we don't really know. From the other side of things, calcium influx is essential for that response. What's downstream of calcium, we don't know yet, but we'll know because it's, one can easily imagine experiment with blocking and manipulating. So far, we don't know.
Great talk. My question goes more like related to that. So what if instead, since your time is really long, what if instead the defect being like activating this interferon gamma response, and it's more like shutting it off, especially because a lot of these interferon gamma genes at the mRNA level, they're really controlled to get shut off. It's really important that you shut them off. And those regulations usually comes at the 3' UTR, which is controlled by RNA structure.
Yeah.
So if you mess around with RNA structure in other genes, like in 3' UTRs of mRNAs, would that affect that time at which you're being in high inflammation? And that might destroy beta cells.
Very interesting. I can't answer that, but the general answer is that nobody knows what the rules It's similar to the microRNA field 10 years ago. Nobody understands the code or the rules of when a double-strand RNA that you imagine, nobody images double-strand RNA, right? You just calculate them based on sequences, right? So nobody knows when a putative double-strand RNA will be dangerous and activating strongly interferon response, and maybe it will be just totally innocent. We just don't understand the rules, right? So when I'm talking with my computational friend there, he is helpless. He doesn't know. He said there are thousands of candidates. We don't know.
I mean, but you can start with those interferon-induced responses and look at the predicted structures and do crystal.
Actually, you have a point, and it turns out that that's an interesting loop that I haven't thought about carefully enough. Interferon-stimulated genes turn out to have double-strand RNA structures in their tail. I'm not sure what the physiological significance of that, but it's interesting. Our approach to that problem is more simple-minded. We're overlapping, beta cells in high glucose produce the danger signal, low glucose they don't, high glucose with nifedipine they don't, alpha cells with overlapping, hopefully we'll end up with like 100 genes, and among them will be the bomb, the time bomb, but nobody knows.
It's very interesting to look at mRNA half-lives to see if the half-life is extended in your ADAR or not, and that might give you an answer of if this is actually happening or not.
Interesting. Thank you. Interesting.
Just one final question. So you showed that nice predictive schematic of these kind of feed-forward loops at the end of your talk. I'm curious if you have any direct or even indirect evidence that if you just enhance beta cell workload or pick your favorite stressor, right? Do you see differences innately in wild type beta cells in the function or activity of ADAR just from the stress alone?
From everything we looked so far, ADAR is immobile, is constitutively expressed in all cells in the same, decent levels. ADAR activity is not changed. The dynamic element is the spectrum of targets. So it's not about ADAR, but it's a very good, experiment in direction. And I think that the clinical hint is that if you lower metabolic load in the honeymoon period, suddenly things get better. Admittedly, nobody opened up the pancreas during the honeymoon and asked, is there less inflammation or just beta cell wake up because they are now less stressed? I don't know, but that's the hypothesis. ADAR itself does not change. It's always the same.
Well, in arguing against that, there's in humans, the data has been clear now that even exquisitely tight control with automated insulin delivery after diagnosis is not associated with preservation of beta cell function compared to those who don't have that. So how do you right size that with your hypothesis?
It's easy. No, not easy. No, so one thing that maybe I didn't emphasize enough. I think there's a consensus that type 1 diabetes is an autoimmune disease driven by autoreactive T cells, right? I am talking here about the first stage, which instigates that, right? An antiviral response that triggers the whole thing, right? I think this relates, I think, to the beta cell injury and the environmental trigger that Des is talking about. And I think this can be prevented. I think once you pass that stage and you have lots of autoreactive T cells, the horses went away already. Now reducing the interferon response, I don't know if this will help or not if you have a bunch of active T cells. So I'm talking about the first kind of innate step of that disease. That is my easy answer to that. But I really. Anyone else would like to ask? That's a question. Is there another?
Oh, it doesn't show you. Okay, I'll ask.
No, that's it. Oh, okay.
Thank you, Dr. Dor, and everyone else make possible is affecting testing program of research. This is the board.
Thank you.
So the view of T1 G parents of T10 Can you please offer your vision for the future? Here is part for this research and work and possible potential you want to support. Then there is the thing is
Thanks, Dr.
as well. It's not related to patient care.
First of all, I'll defer to the SA. Second, yes, support JDRF. These are the angels of the whole thing. This research is about the cause, is about pre-clinical stages, about understanding the genetic and the environmental interface before diabetes. So it's more about the very, very long-term implication. If this turns out to be right, would be prevention, I think, rather than reversal. Other than that, I'm super optimistic about the future, but this is because I listen to him. So
Just leave it at that. I think everyone here is searching for the mechanism. This is what's held up. Efforts to prevent the disease, cure the disease, but we really don't understand the precise mechanism. We understand a lot about the genes, we understand a lot about the immune system. We don't fully understand again why the beta cells. I think this is the search, for truth. And to have people like Yuval working with us is great for all of us. So lunch is observed at the back, and we again thank you for being.
thank you. Thank you very much.